Thank you. Thank you to ARM for the opportunity to present Celyad. We're a company that is focused on engineered cell therapies, in other words, cells that are modified. We're not using non-modified cells in our technology, and we have two platforms, one in cardiovascular that has been basically the foundation of the company. It is a technology that was acquired from Mayo Clinic back in 2007. And it is about transforming cells so they could have a repair mechanism on the heart. I won't describe too much the technology, but it is really a very differentiated approach to cell therapies in the cardiovascular space. Another platform that we have is Oncology that was acquired more recently in January of this year. It is using natural killer CAR T cell platform, which is again very differentiated and very unique. I'll go over that in a couple of slides as well. All our technologies are covered by a broad IP portfolio. We have our own GMP production facility, and we believe that this is also a very important asset for us because in order to manage the cost of goods, and I think someone mentioned that this morning, the know-how associated to bringing those technologies to the patients are not limited only to the IP, but also to the know-how in the manufacturing space. We have a seasoned leadership team, and we are based both in Belgium and we have a subsidiary in Boston in the US. In terms of development timelines, our first program is based on a product that is called C-Cure. We have completed a phase 2 trial in 2012, and we're currently in phase 3 in Europe. We have completed enrollment of a 240 patients trial in Europe. We're hoping to see the outcome of that trial by June of next year. We have a similar trial that should start in the US pending FDA approval, hopefully before the end of this year. It is a very similar trial with a different set of endpoints because the European Medicine Agency and the FDA do not always agree on what endpoints should be used to assess how a product is efficacious or not. On the oncology side, we are currently in phase 1 in two indications, acute myeloid leukemia and multiple myeloma. It is currently running at the Dana-Farber Cancer Institute in Boston, and we're looking forward to expand that to 3 or 4 additional hospitals in the US over the course of the next few weeks and months. We're planning to start other solid and blood cancer studies in Europe and in the US over the next 12 months. We also have an allogeneic platform that is, again, pretty differentiated and unique, and we have two other programs in oncology, NKp30 and B7H6. I will not talk too much about those because those are still in early development. On the cardiology, we are talking about an autologous approach. The invention that was made at Mayo Clinic was if you take cells from a patient and you expose them to a number of factors that are responsible for cardiac development, cardiogenesis in the embryo, you're going to get cells that are more capable of repairing the heart and inducing self-repair. And this is, it's 7 years of research, but leading to extremely interesting outcome in animal studies, followed by a phase 2 study that we ran that showed much improved cardiac function with a statistically and clinically significant increase in the ejection fraction, the same thing in cardiac structure, and also the exercise capacity. The p-values are extremely striking for a study that was relatively small, 45 patients, but that is due to the narrow spread of the data in the patients. Here we look at ejection fraction. You can see that at baseline, the ejection fraction was pretty well matched between treatment and control, which is not always the case in small studies. You can see we have an ejection fraction of about 28, whilst at 6 months, the treatment patients improved by 7 points, which is about 25% improvement. Again, that is pretty good in this type of patient population. The volumes have also been reduced significantly, again about a 20% reduction in end systolic volume, 25 ml, whilst the diastolic volume was reduced in a non-statistically significant manner. The overall heart mass was reduced, and if we go to the exercise capacity of the patient, you can see that it was also improved by 77 meters versus the control. What is interesting about that data is that it is very coherent. Whatever parameter we look at in the trial, there is an improvement of about 20 to 25% of the various tests that we've measured. This is obviously not a guarantee that the phase 3 trial will be successful, because the endpoints are different, but it certainly is a good indication and a mode of action that is consistent in terms of results. We are currently, as I said, in phase 3, progressing well into that trial. Hopefully we'll get the US part of that trial approved by the end of this year. On the oncology, which is the newest acquisition that we have made, we are using a natural killer cell receptor called NKG2D. It is expressed on the surface of NK cells, and it is a receptor that is there to serve as first line of defense when cells get stressed, either through viral infection, autoimmune disease, or tumors. The cells start expressing ligands to NKG2D. NKG2D then gets activated on NK cells, and NK cells come and do their action on those disease cells. The NKG2D ligands are expressed on a very big number of cancers, about 80% of all cancers. You can see here some of the most prevalent one. We talk about ovarian cancer, bladder, breast, lung, liver, etc., etc. So it is basically unlike other CAR-T technologies. This is a CAR-T that weaponizes a T cell to act against a very wide variety of cancers. So we do not need to have an antibody platform to find a specific target for each specific tumor. This would be a more or less generic approach of treating cancer tumors. The data we had at the time of the acquisition were animal data, and you can see here in a mouse model of ovarian cancer, animals treated with the CAR NKG2D T cell versus animal treated with T cells that were not transduced with NKG2D, and you can see a very, very nice response. But I think what is more interesting, because we see a number of mice studies showing results like that, but what is interesting to see is that experience was reproduced in 5 different types of cancers and showing every time exactly the same outcome, meaning 100% survival in the untreated animal and in the treated animal, and 100% mortality in the untreated animals. The other example, the previous slide was on ovarian cancers. This is on multiple myeloma. On the left chart, you can see the same experiment we saw before. Again, 100% survival, 100% mortality. But what is more interesting is on the right side. To our knowledge, this has not been shown before in animals. You can see here I'm not sure if I have a pointer there. Whoops. Yeah. You can see here, those are the surviving animal, and those poor surviving animals, we subjected them again to a new tumor infusion. And you can see that the animals that survived before survived after, while if we inject a different type of tumor, all the animals die. So those animals basically acquired a very specific memory that would fight against a re-challenge by the same tumor type. That's obviously very exciting in terms of mode of action. It calls upon T cell memory and the activation of certain responses. So we also have an allogeneic T cell platform that is currently under development. what we do is silence the TCR, and through competing through the infusion of a molecule that competes with the transduction pathway of the TCR. Our goal is to eliminate graft versus host response, or at least reduce it very significantly, and that would allow the move to allogeneic platform. Now, also we want to be mindful about what we say. Allogeneic is certainly a very interesting approach, but one step at a time. We first need to demonstrate that autologous approaches are efficacious, and allogeneic approach are a next stage development. Thank you very much.